Hello. For those who went to Ivan's talk, this is the headline. This is about a clinical trial of a group of bacteria that were meant to duplicate the effects of a fecal transplant, but the trial failed, hence shampoo washes out. So contra me, I'm going to encourage puns. All right, so this is a story about microbes, and I wrote a whole book about stories about microbes. So I'm going to start by telling you one of them. So cast your mind back about 350 years ago and to a town called Delft in the Netherlands, where this man, Anthony van Leeuwenhoek, is about to change the world. This man is not a scientist, he's not a scholar of any description, but he is a civic servant and a draper, but he does have two qualities that are incredibly important for our story today. One of them is that he is insatiably curious about the world around him. He's very keen on exploring that world with whatever tools he can get his hands on. And the second is that he's the maker of one of those tools, which are at the time the most powerful microscopes around. It looks like this. This microscope is sadly upside down and back to front. But if it were the right way around, then at the bottom of this spike, you'll see a little gap. That contains a beautifully spherical lens, which Leeuwenhoek grounds himself and then sandwiches between these two brass plates to create an object that looks like this kind of glorified door hinge, but which magnifies objects better than anything else around at the time. And Leeuwenhoek turns this thing onto everything else around at the time, onto bits of animals and plants, and into little samples of water that he collects from the canals and rivers of Delft, from urns and vases that have collected rainwater outside his house. And wherever he looks, he sees these things, small animate objects, clearly alive, existing below the threshold of his perception. These are bacteria, microbes. And Leeuwenhoek at this point has just become the first person in the history of all of humanity to see these things with his eyes. And that, to me, is one of the most remarkable moments in all of biology, when we suddenly become aware that there is this whole layer to life that exists below what we can see with our own eyes. And that layer we are now very much aware of. We know of its link to disease. We are increasingly aware of its link to health, about how microbes influence all of these aspects of our biology. Leeuwenhoek wrote to the Royal Society about his discoveries, and by the time he died in 1723, he was certainly one of the Royal Society's most famous and most published members. When he died, he bequeathed upon them this black lacquer cabinet with 26 of his amazing microscopes in them. But for some reason that no one really understands, that cabinet was lost. And still no one knows what happened to it, but the microscopes disappeared. And this was a huge problem at the time because Leeuwenhoek was incredibly cagey about his technique for making those microscopes. He didn't really talk about them very much. So when his gift to the Royal Society was lost, it did leave people in a bit of a bind. It made it very difficult for others to create the same instruments and reproduce his work, which arguably set the study of microbiology back for a while. Now, when asked why didn't you just tell everyone how to make the bloody microscopes, Leeuwenhoek said this: "Not one man in 1,000 is capable of such study because it needs much time and spending much money. And over and above all, most men are not curious to know. Nay, some even make no bones about saying, what does it matter whether we know this or not?" So here is the father of microbiology 350 years ago. He's complaining about money, about public engagement. He has bizarre attitudes about open science. And he doesn't seem to know that women exist. Fortunately, everything is better now. Yeah. So I like telling the story of Leeuwenhoek whenever I do a talk about the microbiome, which is not what this is going to be. But I like telling that story because it has an interesting character who does interesting things. And I think that these kinds of stories matter in science. And I think these kinds of stories are abundant in science. They're all over the place. So those of you who are scientists, these are the stories of your lives. They are stories of triumph and joy, of curiosity and frustration. This is science as it truly is, as a human endeavor done by people who feel things, who have experiences, who are more than just the sum of the papers they publish and the discoveries that they make. But often, this side of science is omitted, dismissed, sometimes even looked down upon as being too fluffy, too hand-wavy, not actually part of the science, and something that that is therefore often omitted in the so-called primary literature, or even in media reports about science. I think that is a problem. I think it's a detriment to the world of science because it disconnects it from this rich humanity that suffuses it. Those kinds of stories are what I relied on when I wrote this book, I Contain Multitudes. Getting people who don't care about microbes, and let's face it, most people don't care about microbes, to read a whole book about this topic is a difficult thing to do. And one very easy way of getting people to care about things that they wouldn't otherwise care about is to get them interested in people who care about those things. And so I Contain Multitudes is not just a textbook about the microbiome, it is also a collection of stories about the people who work in the field and who make the discoveries that allow us to know more about this unseen world, from Leeuwenhoek on to people working today. Now, I stress that not everyone agrees with this approach. So I want to show you all a 1-star Amazon review of my book. Subject cries out for charts, lists, tables, diagrams, footnotes, text. I'm not going to read the whole lot. This person did not like the book. Which is fine, because a book is a piece of art, and art is subjective, and therefore people can totally not like something that I might like. I bring this up, though, because I think that this person dislikes the book for reasons that are dumb, which are the following. Here is what this reviewer believes to be synonymous with science and the conduct of the same and should be the focus of writing of it. Charts, lists, tables, diagrams, details, complexity, and seriousness. And here is what my reviewer deems to be antithetical to science and the writing of the same: feelings and emotions. God forbid we should ever talk about feelings and emotions when we discuss the science. And to me, this is a very bizarre and retrograde way of looking at science. And I think it dismisses all of that very rich humanity that we talked about, and I think it stops people from considering science not only in its best lights as the work of creative, passionate individuals, but also in its worst lights as something that is inherently prone to things like bias and ego and the quest for power or for fame. All of these sides to all of the things that we do as people are very present in the conduct of science. And I think that without telling those stories, we give people a very skewed perception of what science is and, in fact, who does it. I've talked a bit about how powerful it can be to rely on storytelling techniques. I think about this a lot when I think about my audience. So here's an example of my audience. These are just random people sitting on the London Underground. They are bored, they are distractible, they have many other claims on their time and their attention. They may last have done biology at high school. Think about what all of you knew about your respective fields when you were last at high school. They also have this incredible superpower which allows them to nullify any attempts I have to write something that they might be interested in, which is that they can stop reading at any time. And so everything I do when I construct the pieces that I write is partly about gaining people's attention and keeping it consistently on a sentence-by-sentence, paragraph-by-paragraph basis. I also assume that these people do not give a shit about microbes, because fundamentally, there is no reason for them to do so, okay? And what we might ask, therefore, why do people not care about microbes, or really about anything else that we might want to talk to them about? One common answer, I think, is this, that people don't know anything about these topics, and I think this is the wrong answer. This is an answer fueled by the so-called deficit model of science communication, which treats the audience as if it was some kind of empty vessel into which you pour information and facts and knowledge, and lo and behold, they become interested, and maybe they'll change their mind, whatever. This is not how people process information. We know it's not how people process information because this is an empirical line of research that many people have studied, and everything we know about the science of science communication tells us that it's not really just facts or information presented in a vacuum that matter. I think the answer to this question is that people don't feel anything for this topic or for any other that they might not necessarily care about. They have no emotional tether that links them to this material that is unfamiliar to them and not necessarily relevant to their daily lives. And to the extent where people actually do feel anything about microbes, it is that they feel fear and disgust. But you can change that. You can use stories. You can use the stories of people who do the work to replace those feelings with things of, say, curiosity or wonder or just interest. Why do other people care about the stuff that I don't care about is a really fundamental thing to ask about. And because I do stuff like this, because I look for interesting stories of people who do the work, I found that you can make people care in large numbers about things that you wouldn't expect them to care about. Here are 3 pieces that I wrote for The Atlantic that have all massive hits that are about lichens, hagfish slime, and hippo poop, respectively. None of these, if you'd asked me beforehand, I would have suggested to you were going to be massive viral hits, and yet here we are. Lichens was at the top of our website for about 4 days in 2016 over the time of the Republican National Convention. And hagfish slime is still today probably the single most read thing I've ever written for The Atlantic or for anywhere else. So stories can be really powerful. But I think one really crucial thing to remember, which we've heard a lot about today, and which I'm going to talk about for the rest of this talk, is that stories can also powerfully mislead people. And that means those of us who use storytelling and who think about these things have an incredible responsibility to use that power wisely and to think about what stories we're telling, who we're featuring in those stories, and so on. So, for example, who remembers this? Okay, so this is a screen grab of a video that went viral on Twitter at the end of last year, in which a mama bear and a baby bear are climbing up a snowy cliff and the cub repeatedly slips. So it makes it almost its way to the top, slips all the way down to this mountain, climbs all the way to the top again. Bizarrely, the mum seems to swipe at it. It slips way far down, finally manages to climb back up, and the two trundle off into the forest somewhere. And this was portrayed as a beautiful tale of persistence. Why can't we all be like the baby bear? Go, baby bear, you overcome those obstacles. Which was exactly what was happening at the end. No, it turns out that this story has a darker twist to it because it is the story of a reckless drone operator trying to film the two bears and getting too close, causing them to panic, and setting up the circumstances in which this baby repeatedly slips down the slope and almost dies. So what looks like this story of life-affirming triumph and persistence against all odds is actually a story about humans being dicks to animals, which is kind of an evergreen story. Okay, really simple example about what we think we see actually being reflecting a very different narrative than first appears. And this story was at the top of The Atlantic over the midterms, which was interesting. All right, I've written other pieces about areas of science that are nuanced and complex and sometimes problematic in their presentation. So close to home in the area of the microbiome that I've already talked to you about. Much has been said about the effects of things like antibiotics and C-sections on a baby's microbiome. Small pilot studies have been done showing that babies who are born through C-sections or through vaginal delivery end up with different communities of bacteria living on their bodies. Thing is, if you then look at adults who are born via C-section or vaginal delivery, you don't see any differences in their microbiomes anymore. So clearly, these things level out at some point in time. And then the question becomes, okay, but do those differences, to the extent that they exist, for the time that they exist, actually make a difference to a baby's eventual health? And the answer is, we don't know, because no one has done that study. But that hasn't stopped a lot of pieces being published about whether C-sections are bad for a baby's microbiome or so on. I wrote this piece about why those stories are much more nuanced and much more preliminary than they seem to be. Similarly, in the microbiome space, there are a bunch of people who have quite recklessly done their own kind of DIY fecal transplants, which, for anyone who doesn't know what a fecal transplant is, it is exactly what it sounds like. You take poop from a supposedly healthy donor and put it in the body of a sick person, except there are several cases in which people who aren't sick take poop from sources that they haven't vetted and done this thing to themselves for questionable motives. And I've written pieces on this as well. This is an op-ed I wrote for the New York Times about how this idea of a healthy microbiome is flawed, that we still don't really understand how these communities affect our health, much less to get to a point when you can recklessly take someone else's microbiome, put it in yours when you don't have any existing health problems, and know what is going to happen as a result. To do that and to write a lot of pieces about this practice seems to me to be almost encouraging it, or to give a certain amount of validation to the people who are doing it, which I think is problematic. I've also written a lot of pieces in the fields of neuroscience and psychology about areas of studies that have issues with them. So the one at the top is about oxytocin, the much ballyhooed love hormone or cuddle chemical or moral molecule or other ridiculously alliterative epithets. Oxytocin has a huge amount of hype as this driver of all kinds of social positive behaviors, everything from trust to cooperation to morality. The reality is actually very different. The way it works it certainly seems to influence our social lives in interesting ways, but not in clear-cut and not in entirely positive ways. And I've argued in that piece for Slate that the hype around oxytocin is not only a bad reflection of the underlying science, but also potentially dangerous. The bottom two pieces about two other areas, one about brain training games, in which repetitive simple tasks like memorizing lists of numbers supposedly then generalize to all kinds of other abilities in terms of memory, attention, things that then benefit us in our daily lives. The evidence for that is very weak, as Dan and others have shown in a review. And then also studies on the benefits of bilingualism on executive function. So this catch-all term for all these higher aspects of our cognition are supposedly boosted by being able to speak two languages from an early age. And again, the foundational science behind that has a lot of holes in it for many of the reasons that you've already heard about today. Along similar lines, I've done a lot of reporting since 2012, which was, 7 years ago, on psychology's replication problems, this idea that many classic results in this field cannot seemingly be reproduced by people who are trying to repeat those same experiments, and the reasons for why that might be. All of the things that we have heard about already, things about publication biases, about the ways in which researchers can make choices in their experimental design, flexible choices that almost guarantee them positive results without those results necessarily needing to be true. Now, all of these areas have several things in common. They are large fields of research with seemingly a lot of positive supporting work behind them. They've generated dozens and hundreds of papers, and certainly dozens and hundreds of news stories about those papers. And yet they are subject to these kinds of flaws, small sample sizes, playing around with experimental design until you get positive results, publication biases. They are, and yet we write about them a lot. And I know this because I used to write about these areas a lot. So when I started science writing in 2006, I did it by starting a blog called Not Exactly Rocket Science, which I wrote on my own and then eventually became part of Discover and National Geographic. This is the blurb that I wrote in 2006 for the blog. And at this point, I didn't ever self-identify as a journalist when I started. That came later. And this blurb, I think, kind of reflects that, this idea that the blog is an attempt to celebrate science, to make it interesting and fun. All the articles in this blog are based on peer-reviewed research and written from primary papers, not press releases or news stories. And that last bit is still more or less good, and what I still try and do. I don't make use of press releases. I write from primary research. But what we see here is the centrality of celebration as a goal. The whole point of this was to be yay, science. And also the canonization of peer review as a metric of quality. What I am saying here in this blurb is that peer review is the marker by which I'm judging research. And I'm treating it almost as gospel, right? As if something appears in the peer-reviewed literature, I'm taking it as being good. And we know, or at least I hope we know, that's not the case. You've already heard many reasons for why that is, why ostensibly bad research gets into the peer-reviewed literature, why a reasonable proportion of that literature ends up being retracted. But this is not the understanding I had of science when I was starting writing about this, which is why two of the earliest pieces that I wrote for this blog, number 4 and 14, were about papers like this. So the fourth piece I ever wrote for the blog was about a study showing that pictures of eyes in public settings could make people more honest, maybe them behave more pro-socially. 14th piece I ever wrote was about a psychology study about the so-called Lady Macbeth effect, that if you primed people with words that reflect physical cleanliness, that they would also affect their feelings of moral cleanliness. Both of these studies have been discussed as part of this discussion around reproducibility in psychology as sort of flashy effects that that get their way into major journals, that get their way into science writing, but are very difficult to reproduce and maybe don't have the strongest theoretical bases behind them. So I think back on this early work, and I kind of cringe a little bit because it reflects a very naive understanding of how I think science works and what I think good science writing should be. And I feel like I had sort of a come to Jesus moment in, I guess, 2010 to 2012, as I started realizing more and more about issues of reproducibility. So I started realizing that more and more of the papers that I was writing about turned out eventually to be flawed, that were the subject of rebuttals and retractions. So here's where I am now. Now I absolutely do self-identify as a journalist, and this is what I want to do. So not just to tell good stories, but also to unravel bad ones. So our goal is not to just be a cheerleader or a celebrant for science, but also to hold it to account, to also be a watchdog. This means, crucially, that I am not about outreach. I don't do advocacy for science. I don't even really do science communication. Communicating is part of it, but it is also more than that. And so the underlying premise, the motivation behind the work is different. But also, hopefully, as part of that, comes the discipline of a science journalist, by which I mean I try not to trade in snark or gossip. I'm not out for blood, I'm not out to take anyone down, and I'm certainly not an instrument of vengeance for researchers who feel they've been wronged by a rival or a competitor. It is probably going to be useful before we get further down the road with this to show you this diagram that I wrote a while ago, which is every scientist versus journalist debate ever, in which good journalists write pieces in which they're annoyed with bad scientists, but forget that bad journalists also exist and that many good scientists have had bad experiences with bad journalists, as we've already heard about. And good journalists and good scientists forget that there are bad people on both in respective fields and end up annoyed with each other, while the people who everyone is actually mad at aren't listening because they're not part of the debate because they don't care, they are in the pub. Or, I don't know, you guys probably don't have pubs, the bar, or whatever it is Americans do with their spare time. Gun range, I don't know. So as a journalist, here is what I value. These are the things I care about when I'm writing. Accuracy, above all else. I want things to be right. Being analytical, and I sort of prefer that term to either skeptical or critical. Being thoughtful, like actually thinking about the things that I'm writing about beyond just, is this good or bad? Like, what does this mean? And I'll talk about this more in a bit. And above all else, like really being fair. If I'm criticizing someone, giving them a chance, giving them right of reply. Okay, so how does one do all this? I think that we've heard a lot already about like simple tips and tricks for assessing a study, ways of looking at statistics that might be strong or weak. I think these are all incredibly useful. But I would also remind everyone that there is no easy heuristic for judging the quality of a piece of research, and certainly not when you cover as many areas as I like to cover. I write about everything from neuroscience to cancer biology to social science to paleoanthropology. A guide to how to work out whether a psychology study is strong or weak is not really going to help me that much when I'm writing about, say, a new Neanderthal fossil. So you need to have practices that help you work out whether something is strong or weak that work across disciplines. And by far the most important one, I think, as a journalist is to call people. Like to ask other people who are independent from the thing you're writing about whether it's strong or not. And this is just the central act of what we do all the time, not whether by phone or by email, no matter. It's about trying to seek outside expertise, outside independent expertise, and then integrating across all of those opinions to essentially do a form of peer review. And this is also why I disagree with this idea that that science writers should only talk about peer-reviewed science. I write about preprints. I write about preprints quite often, actually, and I think that that is a good practice for science writing. And I think it works because I don't deify peer review anymore. Like the fact that something is peer-reviewed is fine. The fact that something isn't, is it going to be at some point? Is the plan to submit it to peer review, but you're putting it out there now? In which case, fine, I'm going to do the job myself, and I'm going to send it out to people who I think can give me an opinion about whether something is strong or weak. And I think part of this, the really important part of this is to pick your people wisely. So if you're writing about something that's interdisciplinary, for example, you need to get opinions from people across those disciplines. If you're writing about something that is statistically complicated, you need to call a statistician, and so on. If you're writing about something that's ethically complicated, which I'll get to later, you need to call an ethicist. All right, when I do this, what I'm really after are these two things, critique and context. So critique, I want to know whether the thing I'm writing about is solid or not. The method's appropriate, are the conclusions valid? Tell me if I'm about to do a long piece for The Atlantic about something that's actually bad. I do stress that it is really, really important to people to, if you do have criticism, to be honest about it. I really appreciate when people are that. I do recognize that some people are loath to criticize their colleagues in public, and that certain groups of scientists might face harsher repercussions than others for being critical. If that is the case, then at the very least, let me know off the record or on background so that I have some sense of what weaknesses of a thing I'm writing about might be. Context is just as important. So whether a new thing exists in the background of lots of other things that are similar or a bunch of historical research that I need to know about to write about something properly. And that's really important because there are loads of ways of writing bad journalism beyond just writing about weak studies. So here's an example. I wrote this piece about a new computational platform that, to allow people to, who study infectious diseases to analyze the results of sequencing runs and to very quickly work out what's in their sample. There's nothing about this piece that's wrong or inaccurate, but when it was published, it became very clear to me that from the critiques I had of it, that this is just one of many different computational platforms that basically do the same thing that are largely unmentioned in this piece, which more or less portrays this new thing as like the only thing or some big significant breakthrough when it is in fact like one of many. And I don't like doing stuff like that because I feel that I have failed my readers in properly grounding anything that I'm writing about in the context of what else is going on around it. So, you know, lessons learned from stuff like that. I had hints of that when I was doing the reporting and got comments from other people, and probably I should just have listened to my instincts on it because it is totally okay to kill stories, right? Like that is almost the most powerful thing we can do, to just say, no, I think like based on what I found, this is not worth reporting. And you can do that for many reasons beyond just this is bad. Sometimes things are bad in interesting ways, and that makes it worth writing about. But not being able to do your job properly is one good reason. So here, these headlines, none of which were by me, were all about the announcement that Ebola had been discovered in a Liberian bat, which was the first time that virus had been found in West Africa, the source of the largest Ebola outbreak of all time, and that this was important because the exact reservoir of Ebola is still largely uncertain. But this came through a press release that was announced by the Liberian government and a very credible group of scientists. There was no paper. There were no publicly available data sets. There was nothing beyond the fact that these people were claiming to have done what they had done for me to even show to other people to say, does this check out or not? And so I ended up not writing about this because fundamentally I can't do my job if I can't run something past an independent group of people to work out whether to get that critique and that context. Okay, so I ended up not doing this. One really important point I want to make for the next section is that we have talked a bit today about good science coverage and bad science coverage and strong science and weak science. I would encourage everyone to not dichotomize things that starkly. Things are not always just good or bad or strong and weak. Everything contains multitudes. I have regrets now. And so in that gray zone, there are tons of interesting stories to tell. So here's one. This story is based on two preprints by the same group of researchers, one showing that plants can essentially hear, are sensitive to the sounds of pollinators, and another that plants produce noises their own. So both aspects of auditory communication are there in the world of plants. A really interesting thing happened when I sent this out to a bunch of, I think it was like 6 different researchers who studied work in this area, some of whom I expected to be very skeptical about all this, and some of whom I thought might be more sympathetic. Regardless of that breakdown in my head, every single one of them loved one of those studies and hated the other one, which is really interesting because they come from the same group of researchers. So and there is gold there. I think talking about why those two studies got very different responses from different people, regardless of their pre-existing conceptions, is a fascinating tale of science and how it works and how it gets analyzed. Anyone seen Our Planet? Okay, for those who haven't, sorry, spoilers. Okay, there is a scene in Our Planet in which some walruses in Russia climb to the top of cliffs and fall off to their deaths. And I wrote about this, having spoken to a bunch of people who worked on the sequence and some other walrus biologists. And Dan makes some really interesting points earlier about causality, and the path of causality here is really interesting. What the show is claiming is that climate change is leading to lower sea ice levels, which is pushing walruses out onto haul-out beaches in larger and larger numbers, and that overcrowding is causing some of them to climb up the cliffs, and climbing up the cliffs is sending some of them to fall to their doom. There are many steps in that, and some of the walrus biologists I spoke to were really happy with like the first elements, but not really the others. And so this whole claim of is climate change causing walruses to fall off cliffs is not necessarily wrong, but it's complicated. And so unpacking that and looking at how people are understanding different parts of that causal chain is interesting to me. This is a piece I wrote about recent claims about the insect apocalypse. I interviewed May for this. I hope I quoted her fairly. So you may remember a slew of claims a few months ago about how insects were dying in such numbers that maybe insects might disappear altogether within a century. I think that claim is ludicrous, but there is some interesting stuff here about how not just the extent to which insects are disappearing, but also how we make sense of imperfect data. So, and May can correct me if I'm wrong about this, but my understanding here is that there aren't that many good long-term data sets, and they cover a small proportion of the total insect fauna that they are, but those data sets we do have are pointing in an alarming direction. So given that we don't have enough to make representative, super generalizable claims, how do we react to the piecemeal data that we have in a situation where we're actually talking about the difference between not just life and death, but existence and extinction? Again, an interesting way to look at this beyond just, is this right or wrong? Similarly, there was a really interesting debate that came on the back of a science paper that looked at how much of the ocean is actually fished. The original paper said it was 55%. The critics analyzed the exact same data set and came to 4%. And they do that because they're taking very different looks at exactly the same data and ways of measuring fishing impact. And they both have very reasonable claims for why their particular metric and their way of measurement is the right way. And so this piece presents that. And I think, again, the interesting question here is not actually what is the number or like, was the original paper bad or not, but how do we make sense of the world? How do we take a question like this and turn it into an analysis plan? How do we get numbers to answer this question when it's actually not that straightforward? I think about this a lot because our editor-in-chief, Jeff, often says that people don't come to The Atlantic to learn about the news. They come to learn how to think about the news. And that's sort of what I meant when I talked about thoughtfulness being a value that I aspire to, that you're not only telling people what is happening, but you're hopefully giving readers tools to think about the world beyond just like yes or no, right or wrong. Like ways of thinking about and analyzing the news that they're hearing within the pieces that you yourself are writing. So I also am interested in things that take a hard look at entire fields of science and ask whether they're going in the right direction or not, beyond just are things reproducible or is this study right? So here is a piece that I wrote about neuroscience, about which argues that many recent studies, so this recent push to prioritize flashy new tools, often leads us to forget that brains exist in animals, and that those animals behave in interesting ways, and that behavior is a really crucial part of understanding how neuroscience works beyond things that allow us to, for example, track brain activity at a network level or to piece together the firing of a single neuron. This is a piece I wrote about attempts to resynthesize an extinct virus called horsepox. And the ethical question of whether this was worth doing in the first place or not. Again, this is not just about, is this ethical or not? It's also about the fact that the team that did the work got to almost unilaterally make the decision about whether to do it or not. And that's interesting to me. That reveals a flaw in the way modern science is done, in which a lot of research goes on without people knowing about it. And you only know about it after the fact, and you only get to have these ethical decisions after stories have come out, papers have come out, news pieces have come out. Is that a thing that we need to worry about? This is a piece about cancer biology, about how the media that scientists have been using to grow cancer cells for many years, while very easy to make up, doesn't actually reflect the physiology of the liquids in which cancer cells truly exist in our bodies. Is that biasing research in important ways? And finally, there are just areas which are just outright ludicrous, like the announcement late last year in which He Jiankui told the world that he had allegedly created the world's first gene-edited infants, in which a gene that was supposedly, in which he had edited a gene using CRISPR to supposedly make them more resistant to HIV. This has so many problems, scientifically, technically, ethically, in all sorts of dimensions, that I was forced to write a fucking listicle. So, stories matter. Again, I definitely think they matter in terms of talking about science, in terms of showing science as this product of human endeavor. But the story is not just science is great, it's also that science has a dark side. And I know I'm nearly out of time, but you're going to have to bear with me for 5 minutes because this next bit is important. Thank you. All right, so the last thing I want to talk about is who gets to be in our stories in the first place. And a lot of my thinking about this was inspired by and influenced by a colleague named Adrienne LaFrance, who now runs basically The Atlantic, certainly our website and our podcast. She's one of the best journalists I know. And a while back, she did an analysis of her own stories in which she looked at the gender split among her sources for about a year. And she found that only 25% of the people whom she was quoting were women. This proportion just seems to be like a general media thing. Like whenever anyone across different publications has done this type of analysis, they typically find something like 25%. So 3-to-1 overrepresentation of men to women. And what Adrienne wrote at the time was that by failing to quote or mention very many women, I'm one of the forces actively contributing to a world in which women's skills and accomplishments are undermined or ignored and women are excluded. And that world is very much in our faces all the time. It's certainly very much in the face of anyone who works in STEM disciplines. So women face stereotypes about their scientific acumen. They are deemed as less employable or less competent than their male peers. They're paid less. They're invited to give fewer talks. They offer less mentoring. They're subject to ridiculous amounts of harassment and abuse. And and to the extent that any of that applies, it applies doubly to women of color. I always fancied myself as someone who cares about this, so I was very surprised when I did the same analysis as Adrienne and found that I also had the same ratios, 25%, 3-to-1. And this felt just unacceptable. Felt like I was contributing to the same world that Adrienne was describing, and that as a journalist, as someone who works in the media, I am essentially a gatekeeper. My choices of who I feature in my stories influence who my readers see as a scientist, what they think a scientist looks like. And as we heard from May's talk, those decisions about putting people as voices of science have huge impacts often on their subsequent opportunities as spokespeople, as professionals in their own right. So I've been trying to fix this ever since, so I keep a spreadsheet now where I do a running tally of all the stories that I write and the proportion of sources who I contact and quote in my stories who are men and women, who are people of color. That ratio of 25% only took about 4 months to raise to about 50%, where it has stayed ever since. I'm also doing the same, like I said, I'm also doing this analysis for people of color too, who currently stand at about 27% of the people in my stories, which is not bad, but also lower than I would like. We've talked a lot about good or bad science, about statistically strong and and statistically weak science. To me, a science that is not diverse, that is not inclusive, that doesn't care about the representation and the opportunities and the lives of the people who do that work is also a bad science. And I think we shouldn't tolerate it any more than we should tolerate irreproducible work or bad, inaccurate stories. And I think it's important because the people who are in science shape the science that gets done. I think that should be fairly obvious. Again, this just goes beyond gender. This is a story I wrote about an ancient DNA studies about the peopling of the Americas, in which I really tried to make sure that I got voices from indigenous researchers, including indigenous geneticists, to see how their perspectives shape their views on this work. This is a study I wrote about young gender non-conforming children and the decisions that ultimately go into whether they transition or not. This piece quotes several transgender scholars, including at least one transgender scholar of color who is an expert on statistics herself, perfect source. And I've been fortunate to have a lot of people I respect whose work has informed this thinking. I actually wrote a piece once about this person, Ben Barres, who was a Stanford neuroscientist. He passed away, sadly, a few years ago. Ben and his colleagues and proteges were instrumental in showing that the glia cells, the cells that make up about 50% of the brain, not neurons, are and that have long been relegated to being the sort of architectural supporting players in the brain, are actually really, really important for all these kinds of aspects of neurobiology that we had underestimated them for. Ben also spent a lot of time talking in all kinds of talks, even his just basic neuroscience talks, about issues of diversity, challenges faced by women in science. And he knew those challenges well because he was a transgender neuroscientist, so he very much understood the challenges of people at different ends of the gender spectrum. I think about Ben a lot because I don't think it is any coincidence that someone who thought very hard about the problems faced by marginalized groups in science and issues of diversity was also a person who eventually showed that a group of cells that make up half the brain and had long been overlooked and unappreciated were actually super important. Again, I think the people who we are and the backgrounds that we have shape the scientists who we become and the work that the questions we ask about the world, the way we interpret the results of the answers to those questions. And I think that's important. That's why I am trying to make an effort, an ongoing and growing effort to prioritize this in my own work. I think a lot about the work of my partner, Liz Neely, who runs an organization called Story Collider. Story Collider gets people, scientists and non-scientists, to tell true personal stories about science in live stage events. They have made a priority of getting people from different genders, sexual identities, racial groups, people with disability, mental health issues, all people from all kinds of different communities and groups onto their stage to tell their stories, to share the full force of their person on stage. And there was a really interesting small study at a community college where someone got a bunch of students to listen to these stories over a course of a semester, and qualitatively looked at how that changed their perception of what science was and how they saw themselves within it. And many people talked about how they felt a place for themselves in science. They could see themselves reflected in science through the identities of these storytellers. Again, this is why I think that this work is incredibly important, and why I think we should prioritize it just as much as we prioritize being accurate and fair and analytical and all the rest. Thank you very much. Yeah, for sure.
So, you have some questions?
Hi.
So how do you find your science sources, and has the process become any more difficult when you've been trying to bring minorities and women up to a level of...
Yeah, so that's a good question. So I basically just do the same process I use to find sources, but spend more time on it. And that could be like just doing like a simple Google search, like professor and then like the topic that I'm interested in. I'll try and do searches for papers, look at the authors. I'll look on Twitter, see who's talking about a specific thing. There are some databases for sources from diverse groups. 500 Women Scientists runs one that's pretty good. Sometimes those work, sometimes those don't. A bunch of these different techniques almost always yields people. Now, it does take time. It's not a non-zero amount of effort, but it takes much less time than I think people might think. And to me, it's well worth doing. Often I found, like in rare cases when I'm writing about a very specific thing and need a very specific amount of expertise, sometimes there just aren't people of color. Usually there's women. Some very, very, very rare cases there are not. Which is fine, but more often than that, I will be writing about areas that I have covered a lot, that I will count as my beats, and discover people who I've never heard of before, which then begs the question, why have I not heard of them? You know, like what systematic biases existed in the world that I, who supposedly am meant to know this area really well, had no clue that this person even existed? And this is why I think doing this work is important. And also, like, it does get a little easier, but I also try and continually make it challenging. Like, I think there is a problem when, if you're writing about a specific area regularly, to repeatedly just go back to the same person you find there and then, because like usually people from marginalized communities have a lot of shit to deal with, and they're already like burdened with all kinds of things that their people from more privileged groups are not. And so to me, to repeatedly go back like every month saying, what do you think about this paper? What do you think about this paper? What do you think about this paper? Is probably not helpful to them. So every time I write about a new thing in an area that I cover regularly, I always try and find someone new. And there's always someone new. Hi.
Could you hold your question till we get the remote to you so that we can get the question for remote people? Microphone to you.
Hi, I appreciate your perspective on this a lot. I'm a faculty in mechanical engineering, obviously a woman. I wonder if you get pushback on this, and if so, how do you handle that?
I wouldn't say I've had ever any like credible pushback on this. That piece that I put up about I spent 2 years trying to, about like 2/3 of the way down, I actually try and preemptively address the most common yeah buts, of which by far the most common is, shouldn't you be thinking about quality? Like, I am thinking about quality. I'm getting people who are experts in their fields. It just so happens that now half of those experts are women, and some of them, and a quarter of them are people of color. It is interesting to me that this question of why don't you look at quality? It never comes up in the status quo when I'm quoting 3 times more men than women. Like, why aren't we asking questions about whether those people are the most qualified people and all the social and systemic factors that lead to them being the most obvious and prominent candidates while not necessarily being the best ones? So yeah, there is pushback. I would say it has been surprisingly limited. And I think, which is again, I think why it's important for men to talk about this, because we're probably less likely to get pushback on this than a woman making exactly the same point, and that is our privilege and something that we can use to raise awareness of this issue.
Thank you.
Thanks. Anyone else? Hi.
I really appreciated what you had to say about using stories to talk about science, but one of the problems that I've had with that as a scientist is that we often fight against the use of anecdotes to disprove science. For example, anti-vaxxers often use many anecdotes to try to prove their point. And how do you differentiate that and make sure that your stories aren't overshadowing the science that you're really trying to communicate?
Okay, so one really crucial thing I want to say is that I don't think that those two things are separable, right? So when we talk about the stories overshadowing the science, I think implicit in that is the idea that those two things are disparate, right? And that the stories are just a tool that you might use to tell people about the science. I think the stories are the science. I think that the lives and experiences of those people who do research are inextricable from the work that they've actually done. And I think that you can, but I think that it is counterproductive to treat those as things that you can dissociate and for one to just be a tool and the other to be the real stuff. Now, I do get that it is problematic that people use single anecdotes as counter arguments to entire lines of research. But I will say that these things are powerful, right? This reflects how people think about the world. Like one person's story about their child and their experiences as a mother trying to care for that child is more powerful than a meta-analysis or an n equals however 7-digit study, right? Like we can do two things. We can rail against that and despair about it, or we can understand that in understanding that is in itself an evidence-based proposition. Like when we think about communication, we're not just thinking about the things we're trying to get across, but the ways in which we're telling people. And the ways in which we're telling people is an entire field. Like how best to deliver a message is an empirical question to which there has been research. And that research tells us that things like anecdotes are powerful, that stories are powerful, that appealing to people's identities, their values, their experiences is powerful. So we can either I actually think we should use that. I think we should respect it, and we should respect the power of using it badly, and the risk of doing that. But I don't think you can just say that, I think one of the flaws of many approaches to science communication has been to think that we're better than that, or to sort of dissociate ourselves from it. Whereas, in fact, if we actually think about how people process information in other parts of their lives, we all do this. We just need to do it well. Okay. Hi.
I want to follow up on that because I'm a scientist turned journalist, and I do feel compelled to make sure that, I recognize the power of anecdotes, but I do feel compelled to make sure that the anecdote I'm using is kind of representative of a thing, that it's not like an outlier. Right. So, I know you agree with that, but I was wondering if you have a method for how you kind of assess that when you're checking that the anecdote is representative.
I'm not sure. I tend not to I try not to offer strong heuristics for these types of things, because I think that they vary so much on a case-by-case basis. And actually I think sometimes we forget because the most prominent examples of debates and controversies that we deal with regularly, like evolution or like climate change or anti-vaccination, are very stark, right? They have an obvious answer to us. There is consensus. That's very much in the minority, right? Like 99% of controversies in science have no such thing. There's still areas of like active debate. So like actually finding a like, this idea of finding a representative anecdote like does kind of hinge on there actually being a right answer and on you knowing it. And in many cases, that's not the case. So I think like it's about trying, like again, I go back to those values of like being analytical, being thoughtful, being fair, like looking at the perspectives you're putting on a page. Like if you're dealing with an area where there are legitimately two sides, like are you presenting one above another? Like if you're it's an area with consensus, like are you presenting the opposite? Are you now presenting those things unfairly? Like it's a very, very fine judgment, and it's one that you really do need to make on a case-by-case basis.
One more question?
Let's take at the front. Hi.
Hi, so I'm a public information officer at an interdisciplinary research institute on campus, and they talk a lot about team science, and I like to think about team communications. And so when you said that you don't make use of press releases, I was a little bit surprised by that, and I wondered if you could clarify how you view the science communications sort of like ecosystem.
Yeah. I mean, it's tough for me to say because I don't make use of press releases. Like I prefer to go to primary papers. I like it when scientists reach out to me directly, and I find ideas by going to conferences, by looking at debates online, by doing all those sorts of things. Like press releases, by and large, aren't that useful to me. Now, they are useful to a lot of people, so I'm not doing the down or disrespecting the craft of doing a good one. It's just not something that I specifically rely on.
Are scientists connecting you with PIOs to get additional tools to help you tell your stories? Like, are you getting photos or videos or this additional content? Or are you even getting that through your individual reporting?
So often the latter. I mean, videos are not, unless they're like very specifically about the thing that I'm writing about, they're usually not that useful. Images I'll ask researchers for in themselves. I like having that direct line of contact. I honestly think it makes for better journalism.
Okay, thanks.
Thanks.